Hello friends, welcome back to my YouTube channel Nasrin Padasali. So in this video, we'll be discussing the answer for those questions that has been asked under GATE Life Sciences paper. So here I am discussing the question that has been asked in microbiology. In the upcoming video, I do share the answer for other divisions like for botany, zoology and biochemistry. So if you have selected microbiology, then please do cross-check the answers of mine with yours so that if any questions are correlating or it is finding difficult, or if you find some other questions as correct, then definitely we'll discuss in upcoming videos. So first question is that choose the correct match of structural component of bacteria to their function. So this is the first question, or this is any question. If you open Prescott book, this is the first thing that have been given. So they are asking to match the structural component with the function. So first thing is the flagella, which is involved in the process of hemocytosis. So flagella is present in the process of your hemotaxis. Okay, and the cell wall is used to prevent cell lysis. So cell wall is a protective covering of any bacteria. Next thing is the metachromatic granules. These metachromatic granules are acting as a storage hub for this ATP. So they act as a reserve storage for energy and phosphate. Okay, and these magnetosomes are involved in the process of chemotaxis. Yes, these magnetosomes are present few in magnetic bacteria, and this allow the they will take the bacteria. They will these magnetosomes are involved in process of cell orientation by sensing the earth magnetic field. Okay, so the correct options are P2, Q1, R3 and S is 4. I think many will collapse this metachromatic granules with magnetosome. Magnetosomes are involved in the process of cell orientation, whereas this metachromatic granules are present as a storage hub for ATP. Next, the terminal acceptor of electron during anaerobic respiration in methanococcuses. So here they are speaking about anaerobic respiration. If it is an aerobic respiration, then the terminal acceptor will be oxygen. Whereas in anaerobic respiration, substance other than oxygen will be employed. So that might be a nitrate, sulfate, iron, CO2 or magnet. So with respect to methanococcus, carbon dioxide is employed as a terminal acceptor for electron. Next, which of the following mutagen will convert DNA adenine to hypoxanthine? So, they are asking the mutant. So, nitrous oxide is the correct answer. It's a potent mutagen. They act both in a replicating DNA and also in a non-replicating DNA. The mechanism of action of nitrous oxide is by oxidative deamination. So, you should know only adenine, guanine and cytokine consist of the amine group, that is NH2 group. Okay, as a result of this particular nitrous acid, adenine will be converted to hypoxanthine. As a result, adenine will pair up the C instead of thymine. So that's responsible for AT to GC transition. Next, phenolic act as a disinfectant by. So you should know what is the mechanism of action of this phenolic. So this phenolic will act by denaturing, coagulating the protein, and also they will damage the permeability of membrane. So action of this particular disinfecting is by rupturing the plasma membrane, following by leakage of cellular contents. Okay, so this is the correct answer. Next, which of the following methods are used for identification of microorganisms? So ultimately, a 16S rRNA sequencing and percentage of GC content alone is used, whereas the southern blotting and nucleic acid hybridization is not used with respect to microorganisms. So whereas if you take the southern blotting technique, they are generally used to identify the specific DNA sequencing either from blood sample or your tissue sample. Okay, here they are asking what is method used for identifying the microorganism. Next, which of the gram-positive cocci are involved in biofilm of dental plaque? So they are asking about gram-positive cocci only. So generally this biofilm in dental plaque are responsible, they are caused by streptococcus and other species like fusobacterium and actinobacter. So these are anaerobic, but here they are speaking about gram-positive. If you take fusobacterium and gonococcus, they are gram-negative bacteria. Gonococcus is nothing but Neisseria gonorrhoeae. Okay, and the correct option is streptococcus species, that is Streptococcus mutans and Streptococcus pyogenes. Both are belong to gram-positive bacteria. Next thing is the non-suppressor mutation is found in. So non-suppressor mutation, okay, these mutation inhibit the effect of non-suppressor mutant. Okay, so it is found in tRNA. Okay, there are two types of this particular non-suppressor mutant. One is mutant tRNA and another one is mutation on ribosome. Since this mutant tRNA will bind to a termination stop codon of mRNA. Here they are asking where is this non-suppressor mutation is found. So I don't know whether the option tRNA in which this non-suppressor mutation is present or whether it is a stop codon of mRNA. I don't know whether this option is correct or this option is correct. Maybe we can check once the official answer keys will be released. But the correct option is this tRNA, as many things are released, but I don't know. This stop codon of messenger RNA will also be a correct answer. Please do tell me whether you have written stop codon of my messenger RNA or tRNA, like that I don't know. Next, site of photosynthesis in cyanobacteria is, which is a site of photosynthesis for cyanobacteria. So the correct option is chlorosomes. These chlorosomes are involved in a process of photosynthesis antenna complex, and they are generally found in green sulfur bacteria. So here they are speaking about cyanobacteria. So a green sulfur bacteria is again a cyanobacteria. So the correct option is the chlorosomes, and it generally captures the light energy and it will give to wherever it is required. Whereas if you take chloroplasts and thylakoids are not being involved in cyanobacteria, and this chromatophores are nothing but it's a type of cells that are present in animal tissue, surface and animal tissue, that are involved in producing pigment. Here they are speaking about site of photosynthesis in cyanobacteria, which is the chlorosomes. Next thing, match the pathogen with the appropriate disease. So Streptococcus pyogenes, it will cause scarlet fever. So scarlet fever is noted by the bright red rashes, and Brucella species is responsible for causing adolescent fever in children. Okay, and next this Mycobacterium tuberculosis is involved in causing pot disease. Pot disease is nothing but tuberculosis in spine, and your Escherichia coli is involved in causing traveler's diarrhea. So the correct options are P1, Q4, R2, S3. Next, which of the diseases are caused by family of DNA viruses? So the viruses are classified into DNA viruses and RNA viruses. So you should write them in a notebook. What are the viruses belong to DNA and what are the viruses belong to RNA? Only then you can answer this question. Okay, it's the simplest question and it's also a MSQ type of question. If you take influenza, it is a RNA viruses. It is caused by RNA virus and belong to orthomyxoviridae. Okay, and also if you take this rabies virus, it is caused by Lassa virus, which is a segmented negative RNA viruses and belong to Rhabdoviridae family. And if you take smallpox, which is caused by pox virus, and this pox virus is an example of DNA viruses. And also this hepatitis B, which is caused by hepatitis B virus, and this hepatitis B virus is an example of double stranded DNA virus. So correct options are option number smallpox and hepatitis B. Next, correct sequence of overall biochemical reaction which expresses the process of denitrification they are asking. So denitrification is nothing but a chemical reaction of nitrate will convert the nitrate or nitrate by denitrifying bacteria, leading to the loss of nitrogen. So the correct answer is option number this option, that is, they will convert the nitrate, that is nitrite, to nitrate, and it will convert to nitrous oxide, and then it will convert to nitrogen dioxide, and then it will release a nitrogen. So this particular option, if you have given correct, means you will get a mark. This is a MCQ type of question. Which of the following statement are true for archaea? N-formyl methionine is the first amino acid to initiate new polypeptide synthesis in archaea. No, N-formyl methionine is the first amino acid in E. coli. It is not present in archaea. Next, methionine is the first amino acid during protein synthesis in archaea. It is a correct option. Next, cell wall in archaea is contain muramic acid and D-amino acid. No, cell wall is archaea is made up of muramic acid. It is not a muramic acid or D-amino acid. Next, this membrane of archaea contain phytanyl rather than fatty acid. Yes, it is present. Phytanyl is present in the membrane of archaea, whereas other membranes are consist of fatty acid. So the correct options are methionine is the first amino acid during protein synthesis in archaea, and the next option is membrane of archaea contain phytanyl rather than fatty acid. If you take the membrane of E. coli and all, it is made up of fatty acid, whereas this membrane of archaea is consist of phytanyl. Next thing, this is the easiest question. The plasmid given below is digested with restriction in the HindIII and EcoR1. Consider a complete digestion process. So here they had given complete digestion. How many DNA fragment will be released? So they are digesting with both the HindIII and EcoRI. So this is the plasmid map. So there are 4 fragments will be generated. Okay, so you can check this with your friends also. So here there are 2 HindIII enzymes, so 2 fragments will be generated, and again 2 EcoR1 enzymes are there, and it will cut at 2 ports. So in the gel also you will get 4 DNA fragment. And next question is, which of the following are present in gram-negative bacteria? So gram-negative bacteria, your endotoxin will be present and periplasm will be present and liposaccharide will be present. This endotoxin is present in the lipopolysaccharide of gram-negative bacteria, and this periplasm is nothing but a space between the inner membrane and outer membrane of gram-negative bacteria, and this lipopolysaccharide is also present in the outer membrane of gram-negative bacteria. Whereas this tecoic acid is not present in gram-negative bacteria, it is present in gram-positive bacteria. Since it is an anionic protein, it is present only in gram-positive bacteria. Next, the antimicrobial activity of vancomycin is due to. What is the mechanism by which this vancomycin antimicrobial activity is done? Means it will inhibit the cell wall synthesis. So generally this vancomycin will kill the bacteria while disrupting the bacterial cell wall. Okay, so this is the action of vancomycin. So thank you friends, thanks for watching this video.